10, so we may as well start. Does anyone have any announcements? No. Okay. So I'm very pleased to introduce Sally to talk to us this afternoon. So Sally did her PhD with me, and she is now teaching in the biology department at Augustana. And as I was thinking about my introduction, I was thinking about Sally, and there were a lot of things that came to mind. I think that many of you who know Sally, you know that she's a go-getter, right? She sets her eye on something and she goes after it. And she's not afraid to knock on doors and to see if they're open. And because of that, she has had so many opportunities from that. A good example that I can think of is she went after a scholarship or a fellowship with the international office at APS, and so she got awarded that. And so after her postdoc, she spent 6 months working on, after her PhD, she spent 6 months as a postdoc, and she worked in East Africa, where she and some colleagues there wrote a case study, and they also did some extension and some workshops out there. And it just shows how entrepreneurial Sally is. And she has so many wild and wacky ideas, but it's awesome, right? She really thinks outside the box. The other thing about Sally is she has endless energy. I don't know where she gets it from. She's a mother of 2. She had ear while she was still doing her PhD, and I don't know if it was as a result of that, but she kept some crazy schedules in our lab. Do not ask her what hours she worked, because they were pretty crazy. But anyway, it's been really fun. We loved having Sally in the lab, and we had some good laughs. And I think back over the many memories that I have. The one thing I remember about that APS fellowship was that it has caused us numerous nightmares with Iowa State accounting. So before you decide to pursue something like that, talk to your advisor. Anyway, so I'm thrilled to have Sally here today, and she's now teaching at Augustana University in the biology department, and so she's going to be sharing some of the really cool stuff that she's been doing there, introducing plant pathology, the coolest discipline in biology, to people who've never even heard of it. So, thank you, Sally.
Thank you. Okay, if you're staff and you're a professor, this is the time where you move out and then the rest of the people can stay. The reason I say that is I teach every day and I talk to people, but I haven't been this nervous since I had to do my prelims. And then the other time I was this nervous, they said, okay, Sally, can you tell us what you did at Iowa State and present about your research? And then all the other professors came and it was like, oh my God. And so this is the next time, because I feel like, oh no, now this is another defense because my professors are here. And I told the grad students during lunch, like, I wish the professors wouldn't be there, then we would just be okay. But they're here, so we'll make it work. So I teach at Augustana University. It's a smaller Lutheran school. And so I was a bit nervous about that. It's like, it's a Christian school and you're going to teach biology, and how is that going to work out? But it turned out okay. It's a really, really good department. Compared to Iowa State or where I went to school at Egerton in Kenya, it's way smaller. We have 1,800 students total in the whole school, and we have 200 students who are biology majors. And so that's one of the bigger departments there. And then we also offer service courses, which means that we offer courses for the nurses and courses for the non-majors biology, so they're all trying to check a box in there, like we've already done our science course. And so there's all those people who don't really want to be there, but are held ransom and need to take a biology class. I totally understand and empathize what most of you are going through right now with the move. We have a beautiful new science building. It is the heart of Sioux Falls. We like to pretend everybody driving around there can see us do our research that we teach undergraduates, and there's a few people that have come and said they came to Augustana because they saw the science building and they wanted to come there. But when I got to Augustana, the science building was not complete. So the first semester we were in one half of the old building, and we were there for a couple of weeks. Then we were moved in the next semester to another building for a couple of weeks. Then we were moved back to the brilliant, beautiful new building. So it was my third semester in a third office, 3 semesters, 3 new offices, and I had no idea what was going on. I work with some really, really wonderful people. When I was in Allison's lab, it was a female-heavy lab. We had Mauricio and John, and not very many other people there, but they managed with us. So when I got there, which was a female-heavy department, there's a few gentlemen I'll show you, but they're really, really awesome. They're really, really smart. We team teach the courses, so for somebody who hadn't taught and really wanted to teach, that was a good fit for me, because before you actually teach any class, you've listened to it, you will go to class and you will see somebody else teach it. We teach intro bio for majors and intro bio for non-majors. So for each of those, you have an opportunity to see a flipped class and to see a normal class before you actually have to teach it. And if you're team teaching a course with someone, you have to be there and see them teach. So that's been very good training for me. While I was here, I took one random class that was offered in horticulture about teaching, and I wish I'd listened lots more, because it's proving to be the most important class right now. But they are a really, really good people to work with. So part of what I have tried to do is to make a plant pathology package for the non-majors biology class, and so that is the students who are going to be journalists and musicians and government and business administration, all those people who end up in our biology class who don't want to be there, and you're trying to tell them, so this is what I did, and it's really, really important. You need to pay attention to this. They don't hear. And then the majors want to be doctors and nurses, the exercise science. Those are the things that they are interested in. And then I spent a lot of time in the greenhouse working on Goss Wilt, even though the paper was rejected a few times. It's a presentation for another day. So using that information to develop a teaching model where the undergraduate students can be able to create research experiences for those students using Goss Wilt, which is what I spent a lot of time doing for the students. So at the beginning of class, we have an introductory class, especially for the non-majors, and we talk about what a wonderful department the bio department is, and we show that slide, and then we talk about our families, and I talk about my children. So this is Egan, and he's in 6th grade now, and he's my master's baby, and so he's been with this journey for a while. And this is Ia, and she's my PhD baby, and she was 5, that was first day of kindergarten. So they've walked this journey. My family and I, we like to travel, so we're trying to cover all the states, right, in the US. We are at about 30, so hopefully before we leave, we'll get to all 50 of them. And this is APS, that is the society that I belong to. There's about 5,000 of us. I'm very passionate about it because I'm a plant pathologist. And then I explain what plant pathology is, and I tell them my name, and I tell them that I went to Iowa State. So imagine 3 weeks down the road, you introduce yourself and you told them all these wonderful things about them. And then you have a bonus question for the students. Where does Dr. Mallowa come from? What does she like doing? Where did she go to school? What is her specialty? Where does she come from? Can I have a thick accent? I'm probably African, I look African. So they'll throw it in there. She's African, she's from Kenya, she's from Nigeria. Usually they don't get the Kenya right. They'll get something in the ballpark, but at least they'll say something about Africa. They will remember that I come from Iowa State, and I went to Iowa State because there's several people that come from Iowa. They retain that. They remember my children. She's got, someone might throw in a plant, a few people, not very many. And so what usually comes is somebody says, something to do with homicide, and I was like, homicide? Like, really? Yeah. And paper after paper after paper, nobody retained that information at all. they didn't remember what I did, and they absolutely didn't care, because you explained, but nobody remembered. It's like, how can we engage these students to what you do? And my first semester, some of the reviews I got was, oh, I failed the class because I couldn't understand her accent. Yeah, really. You failed the class because you didn't work hard. Somebody got an A in this class. But there was all kinds of excuses, and living in the times that we live in, if you don't talk about it first, then you have to excuse yourself later. So it's, how can we make a package where we will have plant pathology, you will appreciate the fact that I'm African and the things that I do. We have labs for these students, and the way the labs were set up before was, the first lab was about microscopes. We gave the students microscopes, and we taught them about the cell. They are non-majors, so they would be like me today, Yes, there's a microscope. What am I supposed to do here? And it's just overwhelming for a non-major to come into a lab, and that's their first lab. It's like the third day of school. But that's the way it had been done for a long time, and that's what they did. So when I got the opportunity to teach it by myself, I used the case study, and this is a case study that we developed while we were in Africa during my postdoc. So the case study is a cassava case study. So cassava is a crop that's grown in Africa, in Asia. It looks like a sweet potato. It has this big root. And we give the background, and we make the students use that case study. They have to go to the APS website and click on there to find the case study. So they have to see some of these things. Anyway, so they are introduced to a crop that they are not familiar with. Some of the background information that they need to use to prepare for this includes, they need to listen to broadcasts about the crop. They need to listen to broadcasts about the diseases that affect this crop, and those are spoken by people in Ghana and Nigeria that have all different kinds of accents. It wasn't planned like that, but that's where the problem is, and that's where the people are. So they are introduced to that. The case study is based in Uganda. It is based in Soroti in Uganda, where Kevin and Martha, who are friends of mine, come from. And we contextualize it in Africa, where in Africa it is, where in Uganda this disease is. And then we explain to them the situation that is in Africa, for peasant farmers, not for the rich people in Nairobi. This is what the situation is. This is what happens when you get a plant disease on an important crop that is your staple that you need to eat tomorrow. And you contextualize all the problems that are there, and then you allow the students to put themselves in that situation, and then we have discussions. So because we have made it a requirement for them to prepare, they're familiar with the disease, they're familiar with the situation, you've explained it to them, then you let them have an opportunity to be able to build on that and talk about that. We have students from theater that go to town with a case study. They take up roles and they are arguing with each other. And when they are given the information, they really, really own it. And so it's like, what are we going to do with the crop now that we don't have food? Oh, we're going to get money from the government, and we're going to get money from FAO so that we can get more crops. And what I find very interesting about that is students in America, students in Africa get the same conclusion. Everybody wants to ask for aid so that they can solve the problem. It doesn't matter where they are. That is the situation of the problem. And you're like, right now, when you don't have food to eat, how is the aid going to help you as you wait for the aid to come? So we really have good conversations, and we change it around so that this is the first lab that they do. And so because we have contextualized it, they can understand where I come from, and they ask honest questions. So are your parents hungry? No. Is there food? Is that a situation your family goes through? And all the legitimate questions. So we are done with that, and so it gives you an opportunity to be able to talk about the situation. Now we've talked about a plant disease, we've talked about a real problem, there's a problem somewhere that affects real people, you've seen articles about it, and they can relate to those to that situation. Still, some people don't get it, but way more people can remember what I do. After that, we have 12 more weeks of doing biology labs, but they remember what plant pathology is, and they are less afraid of it. So that's been one way that we have engaged the students, especially the non-majors. The second thing that I do is that I ask the students to do an email assignment. It's called the biologized email assignment. So at the beginning of the semester, I tell them that every week you need to email me something about biology. If you looked at your egg and you thought it looked like a cell, you email me and tell me about it. If we said something in class that went over your head, you email me and tell me about it. If we said something that you found really cool, tell me about it. If something is going on in your family or anything as long as it's related to biology, you tell me about it. Part of the reason I set up this assignment is because the examples that I have to give that I think about instinctively are not things that my students will relate to. And I was just telling the grad students, one of the examples that comes to me instinctively is, you're talking about water and how when you're walking on sand and then you walk in the water, the sand is warmer and the water is still cool, and you're giving an example and you say, like when you're walking in the coast of Mombasa. Right. Like when you're walking the coast in Florida, something that the students can relate to. When they sent me the emails, I have learned so many random facts that I wouldn't have learned otherwise, because I have to read through their emails and I learn things about the American culture or about the experiences that they're going through, about who learned about mitosis and how that's important for cancer and they never knew that. And so many really good experiences. Or they see a cartoon or the video that went viral about a student talking about biology, and even students who've left the class will email you about it. Guess what we saw? And it's a way to keep them engaged. Or they got a pet snail. This was Humphrey, and a student sent me an email about their pet snail. So it's a way to engage the students, to force me to learn about them, and to also learn so many things that I would not do otherwise unless I had to. So I feel it's a way to engage them. They're forced to think about biology, and the interesting thing is, because they know I'm a plant person, they try to tell me things about plants, or to research things about plants, or to research things about cassava further, because we talked about the cassava case study or things that we have talked about in class. So I feel like the case study has been important in terms of engaging the students for that, and the biologized email gives them an opportunity to solidify and to share the things that they learn. And also when they're struggling, that's a really, really good way to know they didn't understand what you said, or you're going too fast for them. It gives them an avenue to be able to talk about that. So at the end of the semester, because we are surviving this class, nobody wants to be there, we take a picture and we say, you survived biology 110 with Mallowa and Lewis, and we send the picture to the students, and they're excited about it, and I'm also excited about it. And if you can get a few smiles, even from the people who will be back next semester. Yeah, so that was my semester last year. We haven't survived yet. Next week is picture week, so we will take the pictures. Then the other course that I have had to teach this semester is a course in my area of specialization. There is no team teaching in that course, because it is your area of specialization and you're the only plant pathologist. And nobody wants to do this course. They're also checking a box because they need to do an ecology-based course, and that is where the plant part course fit in. So trying to sell the course is like, who's going to take your course in plant pathology and they're going to nursing school or to medical school? I know. So it was, how can we make it so it will be interesting for the students so that they want to come to this class? So we say it's plant pathology and global food security. We're going to talk about plant pathology because who had a meal today? What did you have? Corn. That's a crop. Thank a plant pathologist. And we introduce it like that and begin to talk about why it's important to the name, even if it's a small part of the course. Then we had more students. So we were very excited about it, the plant people, because there was plant pathology and neurobiology and ecology, and the plant class was the first to get full because it was plant pathology and global food security. And so we had to add that. And then other components that we had is we had the students do a professional interview. So I threw out a couple of names out there Anne Videvar and Steve Lindo and some of the people that I've been honored to work with. And you choose one of these people and make it your responsibility to find them and conduct an interview and ask them about what they did in their research and how that is important for food security. And all those people are probably plant pathologists, the names that we had out there. And some of the feedback that I had from the students, it was a really challenging exercise to even get the people because they are busy people, and that's part of your assignment. I'm not going to email them and ask them to talk to you. You find a way, you know. I just let them know my students will email you at some point. So some of them had to work really hard, like the students who wanted to talk to Anne Videvar, and she's retired, and they emailed this other person and this other person, and can you help us? Can we get, you know, so I always said, okay, I'll help you a little. And I got APS to give us some contact information. But that is one of the things they did, and that exposes them to plant pathology as a field, and it's something that they were excited to do because it was something different. But this person will talk about what they did in their research, which is in plant pathology. Then they had a service project. You had to choose something related to food security and do your service project. And they had a collaborative project, which I'll talk about in a little bit. We did assignment with students in Malaysia and students in Makerere University in Uganda. And that's been the most challenging part of the semester. And I have been telling them, you know what, it's not because you're students, right? Because earlier in my career, when I was way younger, I had to work in a multi-institutional project, and I worked with people in America and in China, and I was in Africa, and I was female, and I was young, and everybody was older and male, and I felt very intimidated, but I never got response because I was just not important. And so the students don't also get response from their other students, and they are very frustrated, and it's like, that's the way these things work. Even when you're in the same country, it can be a little challenging when you have a collaborative project. But it's been part of the interesting thing to have that to do. They have an independent research project that's based on Goss Wilt. We'll talk about that in a little bit. Then we have the normal plant pathology lectures. There's nothing we can do about fungi. We just have to hit it, and bacteria. And so we just do regular lectures. And then we've had laboratories, for which I thank so many people in this room. I came here to beg and plead and borrow because I was teaching a class that didn't have budget. And they didn't, at that time, we didn't know whether I could stay or not. And the department was not willing to invest in buying things that only you can use and then you will leave. And so those labs have been successful because I went to Iowa State and I could come and borrow, and people were very helpful. So we had really, really good labs. Then the global food security part, which the students really, really have enjoyed. We had sessions that I led, that is the IL, instructor-led sessions. And then we had students' presentations, so they chose different aspects. One time we talked about diseases, and they chose different diseases to work on, and we had groups of students present about cassava mosaic disease because they know I like that, and they are after my own heart, and corn diseases. And then we have group discussions about biofuels and how that threatens food security, and the farm bill, because they're totally unaware that the farm bill is not something for farmers, it's something that affects everybody. And so we've had some really good discussions, and we do that a couple of times a week. So it's like 2 courses in one. 2 times a week we have lectures, and it's normal lectures, and then every Friday we discuss the different issues that are related to global food security. So when I was here, some of you will remember that slide. I stole it from Alison. And we talk about Goss Wilt and how it's an important model plant pathogen because 1, the instructor understands what's going on, and 2, it doesn't disappoint, right? If you inoculate, you will get disease, and when there's disease, you know there's disease, and then that's what it looks like. That's what I tell my students. They're going to be doctors, so they have no idea what you're talking about. So when they're diseased, what do they look like? Really. And what I was working on for my PhD was whether we can get infection without severe wounding. And so we were able to talk about what the growers were seeing in the fields and what the researchers could see. And then my research showed, not really showed, but hinted that you can get infection without severe wounding. And we had a GFP from Gwen that we were able to use and work with that didn't cause infection. So now, if I want to use this as a model system, I need a GFP that works, and I was on the 3rd floor, not the 4th floor, and that will soon be a thing of the past, but those of you who understand what that means I cannot do that. Yeah. I needed to talk to the students about that and talk to a colleague of mine who is with bioinformatics, and she was on the 4th floor, wherever she was. So we wrote a grant together, and our grant was to continue that work which Sally was doing for her PhD, because it's been shown in other systems, and to see whether we can do that system for healthy plants. So Emmanuel, who you know, got me a strain from South Dakota because I don't have a permit, and Allison couldn't give me one. And so we did studies in the summer. We got paid for it to do that, like 3 plants, and show that we have disease. So that was exciting. You know, I can do that. I can show you we can get disease. And then she did that part. So she got a construct, and then we were able to do electroporation and be able to see to see whether we could get transformation and whether we could develop a protocol that we could use in class to be able to do that with. So using Goss Wilt as a model system for us to be able to use in our plant pathophysiology class. So can we use Goss Wilt to show Koch's postulates, right? Can we use Goss Wilt to talk about the different microbial features? Can we use it to do transformation, to do the microscopy after the transformation, to teach confocal microscopy, epifluorescence, and things like that? And can the students develop independent projects related to this? Can they develop a hypothesis? Can they analyze data? All the things that you require from upper level students using this particular pathosystem. So these are some of the tests that the students did, and this I pulled out of their reports that they gave, and they could really went to town with it and understood what we were trying to use. So when we tried to talk about the different characteristics of gram-positive and gram-negative, I think it all went over my head until I had to teach it. And like, oh, what's really going on there? But Gwen, I didn't do too badly. So we use that, and we use that as a model system, and they are comparing everything against the Goss Wilt pathogen, and then we are determining Koch's postulate using this same pathogen. So can you get disease? What does it look like? You will come back and you will check on your plants and see what disease you get. You will isolate, inoculate, all the things that go with that. We learn about bacterial streaming. We're still using the same pathogen. We're talking about gram-positive and gram-negative and doing gram stains, still with the same pathogen. When we did the transformations, what does it look like with your control? What does it look like with the transformed bacteria, with the non-transformed bacteria? Does it grow? Does it not grow on the different types of media, with an antibiotic, without an antibiotic? Still, all these experiments, we're working with the same model. So it's like all through the semester, you're talking about the same thing, and so the students kind of get a handle for what is going on. So that when you finally tell them, now you need to do independent projects on this system, they have understood the background, and they have understood what the control looks like. This was looking at the controls for the transformed versus the non-transformed. We're still working on that one because I'm not very. We were able to get Goss Wilt grow on a plate with the antibiotics. So we know there's something that happened that's positive, and we are able to get inoculation, but we were not able to see everything was glowing, so we were not able to demonstrate that we actually had, and so that's still a work in progress, and the students are working on it as well. So this year it didn't work, but next time we have another class, hopefully we will get that to work. With the independent projects, we have the students now begin to use the different skills that we want them to know using this tool. Can you get plant incidents, leaf incidents, the different tools with microscopy, bacterial counts? They have to come up with their own hypothesis, use of one of these ways to collect data, and yeah, those error bars are still giving me grief. But can we analyze the data? Can you find help if you need it? And so that has gone really, really well. I'm really excited about it. It's not all the data that we want or we would like, but for a first time, I think I've been able to make a small bit of progress and start the processes that I would like. And it's something that I'm excited about because I really understand that patho system well, as opposed to if I tried to work on any other patho system that I didn't understand as well. So I feel like it's something good that came out of my PhD. It didn't just go somewhere in a corner. It's not level groundbreaking research, but I'm still using the information that I have, and I am an expert at this particular thing. I spent a lot of time working on that. The multi-institutional collaborative project, we have the students, and we want them to talk about climate change and how it affects plant diseases, and if they choose a plant disease that they want to work with, and they also choose an aspect of that plant disease that they want to talk about and how it affects food security. So at the beginning of the semester, they assort themselves into groups. And I know you can write a report. Everybody knows you can write a report. But can you write a report with somebody else in another country on a different time zone when they do not reply your emails without giving back up and writing the report yourself? So the students from Uganda that we work with are specifically, they have a course in soil microbiology and plant pathology. My students have plant pathology and global food security, and the students in Malaysia have a totally different course. They are doing crop production, but they are all a little bit related. So the students have to spend time getting to know each other, what is important in your university, what is important about the things that you do, and then get together working on the project. We had problems getting them onto our system so that everybody could see each other. Then they gave up on the system, and now they work on WhatsApp. But when they don't reply, then they need to send the WhatsApp to you so that you can get help. But it's been a very interesting exercise for them as well as for us, as instructors, in terms of getting to know each other and seeing how we can use the resources that we have. And then we met because she wanted information about my case study. And I was like, why do you want information about my case study? I actually have a different case study and I have a class. Do you want to work together? And so we got to work together. It's been a good experience. And Mark, it would be nice if we had more information about how instructors trying to do things like this can work together, and rules for how to do that, and protocols for how you can do that, because you have to go through the bureaucracy of the system. If you ever want to use the data, you have to get institutional board approval, and those are things nobody tells you. So you could get on board informally and then have to backtrack once you have the data that you
Exactly. Then you know when you propose these ideas yourself, you know who ends up doing it.
Well, I would say that. And these are my wonderful students who got me a job because they took my class over the other classes, and we had to cap the class because there were too many. And those are the guys, and they were trying to stand like the girls because that's how we stand. But they are really amazing. I had 18 students take this class, and of the 18 of them, 6 of them are going to med school, and they do see that it will be useful to them. If they only have to say during the med school interview about their really most frustrating, challenging experience was trying to get this project done, that's been very stressful, but they've gotten the paper written. This week they need to do presentations together. You have to make the slides together, and everybody presents in their own country. The 3 instructors grade your reports, and then we put it together. So it's a huge component of the grades for my students, and I think it's been challenging, but it's been worth it, at least for me. Everybody is recruited into my class who can help. Xiaoyu, Clarice, my husband, anybody who knows anything about plant pathology has helped me this semester, and I am very, very thankful. The other activities that I am involved in, this summer we have summer camp at Augustana, and so I get to do a session called Plants Get Sick Too. and I'm excited to do that. It's for 3rd to 5th graders, and this semester I will not get help, but in the future, the students in my class will help us to develop what we will do in the summer with the students for camp. There is a South Dakota Youth Institute with the Wild Food Prize. It is nothing as glorified as the one in Iowa, and there was 14 students, but it was very exciting to be able to go there and to talk about the different experiences and to share that with them. This summer, I get to go to Kenya. There is in the largest slum in Nairobi. It is called Sioux Fall. It is a self-help group for communities. So self-help for communities, and it's out there in the slum area. The largest slum is called Kibera, and they started a school out there. And so because we will already have the summer program, when I go to Kenya in the summer, we get to run our summer program with the kids out there. And so it'll be interesting to see how that works. Then they have just started spring break programs, even though we don't have spring break in Africa. So when students in the university are at home during the holiday, then they go to help the different schools. So there is is she my friend? Yes. A friend of mine or a colleague of mine who runs a program in another part of Kenya where she has students on spring break come to do that. And so we would like them to get them to do some of these plant path things where she is she's a postdoc at Auburn, but she runs this program out of coastal Kenya. And so hopefully I will be able to go there in the summer and run this workshop with the students that will be on spring break. I got an invitation because of the work that we did in 2015 to go and work with brewers and tell them about cassava. And so in the summer, I'll also do a workshop with some of the brewers. My big goal that I hope to do eventually is to do a study abroad course that goes to Africa, sort of similar to what we have with the global resource systems here. They don't go to Kenya because Kenya is on a terrorist list somewhere, so I can't take my students to Kenya. But that is really the goal that I would like to have. If we could have an interdepartmental course that would cover all these things and get to go, that would be something I'd be really interested in. That's my long-term goal. It won't happen anytime soon. Then the other thing that I do that you are welcome to join me in doing is that because my dad graduated when he was 86, because he hadn't graduated when he should have, is we started as a family an award series, and because to celebrate that, and that was around the same time that I graduated, so 3 years ago. So the first year we had 3 students, it was just me, and I was broke and I was in grad school, so we gave them $200, a little less than $200 for the 3 students that was my parents at that first award. In the second year, I took my kids with me, and we had 4 students and a little more. Then in the third year, we had my students with a Tri-Beta club and biology club, and I think we had 21 students. And then this year we had a total of 46 because we had the main awards that we started for my family, plus other awards that people can start in memory of their sisters or their loved ones that they care about. And so we had 46 awards and I think $1,800. So I feel like we have grown, and that's something I'm proud of. It's over there, and it's really, really awesome. So thank you for listening to me, and I'm really happy to be back.
Questions?
So do most of the students at Augustana go on study abroad programs?
Yeah, not, I would say a good percent, about 40% actually go in different study abroad programs. In our department, it's way more than that. I would say maybe 60%, because the department has a course that goes to Guatemala and Belize to do tropical ecology. They have another one that goes to Florida to do marine ecology. So it's and that counts as your option, your ecology option. So it's a good number.
So if you develop a study abroad program, will the college be very supportive?
Mm-hmm. Yes, they will. The students want to, but I only know Kenya. Maybe I'll need to broaden my horizon.
So what advice would you have for graduate student, let's say in plant pathology, just to pick on subject at random, that wanted to have a partial or complete career in teaching, and they're thinking about how they can make themselves competitive, what would be your advice?
My advice would be to put yourself out there and do the non-traditional things, because in the end they count more. Everybody has a degree, and so you have to do your degree and do it well, but you also have to do other things. Volunteer to TA if you could. It counts for a lot, and do different activities. I have felt that working with people within APS, even in different capacities, has increased the number of mentors that you have, because you hear different perspectives from different people all the time. So being involved with APS has been a huge benefit for me, that I would say. If you can't, put yourself out there and do this.
Can you share some of the experience, like when you become independent, researcher or teacher, how you collaborate with other people compared to when you were a student?
I don't know whether for me it's very different, because I just ask. I'm always asking for things, but I think you have way more responsibility when it's done with you, and you need to be willing to put in the work. So like with the collaborative project, I had to approach my IT department and get the system working, and if they don't have time and you want this to work, like you need to be willing to put in the work, compared to when you're a student. Hopefully your advisor will put in the work. Yeah, but I would say you have to have way more responsibility and be willing to go the long haul.
I have a question, Sally. So you were talking about how you do the Casavis case study with the non-biology majors. They have to go, it's a flipped classroom, right? They have to go and do all the preparation and then come in. And it sounded like you got, that they all went and did their homework and then came to class prepared.
Yes, they did, because
How did you manage
there's
that?
points associated with that.
Okay.
Before we start the lab, first thing when you come, we're going to take a quiz. And it's on the material, like names of the people in the case study, like very random specific things that if you didn't watch the video, you wouldn't know. And it's the first week of the semester, and they're non-biology majors, so they're so afraid that first week will read it. If you tried it later in the semester, it might not work as well.
Okay. What other questions do you have?
I was wondering where the students were coming from, and if this is a private college, so are they more motivated than maybe a university that would be lower cost?
I really don't know, but the students in biology are very motivated, just because that particular department has a reputation for sending kids to med school. So it's like, it's not true, but they believe that if you go to Augustana, you'll get into med school or whatever medical career path you want to go. So those particular students are very motivated. But whether they are more motivated or less, I don't have a point of reference. So they look pretty motivated to me, but I don't know. Our international students, though, there's a lot more work, because it's a private college and they're rich kids.
Thank you, Sally, very much. And